Sexual reproduction. Sexual reproduction in bacteria. In bacteria, true sexual reproduction is absent. Then why it is called sexual reproduction? Why true sexual reproduction is absent? No new bacteria not at all formed. New bacteria not at all formed by this method. Then why it is called sexual reproduction? Actually, what happened in sexual reproduction? That the genetic recombination, transfer of genetic material that occur in between the two bacteria. So it is called sexual reproduction, but new bacteria not at all formed. How this genetic recombination occur? How this genetic material transfer that occur? Based on that, this sexual reproduction is of two types. Sexual reproduction is of three types. One, conjugation. Transformation. Transduction. Conjugation first observed in E. coli, Escherichia coli by Lederberg and Tatum. Transformation first observed in Streptococcus typhimurium by Frederick Griffith. Transduction first observed in Salmonella typhimurium by Lederberg and Gender. Based on how the genetic material transfer, how the genetic recombination occur. Sexual reproduction in bacterium is of three types. Conjugation observed first in E. coli by Lederberg and Tatum. Transformation in Streptococcus pneumoniae by Frederick Griffith. Transduction Salmonella typhimurium first observed in that one by Lederberg and Gender. Transfer of genetic material in between two bacterium occur by direct contact, conjugation only. By direct contact to bacteria. If the genetic material transfer occurs, that is conjugation. Transformation of genetic material with the help of virus by transduction. Through medium transformation, we will discuss that in detail. First observed by Lederberg and Tatum in Escherichia coli. Here two strains present. One bacterium, another bacterium. Main genetic material. One bacterium consists the plasmid. In another bacterium, plasmid is absent. Bacterium with plasmid, F plus strain. Bacterium without plasmid, F minus strain. So plasmid with fertility factor. Plasmid without, plasmid not, no plasmid, no fertility factor. So F minus strain. Sex pili present in F plus strain only. It donates this plasmid to the F minus. So F plus strain also called donor cell. F minus strain also called recipient. F plus strain that consists the sex pili. So donor cell only consists the sex pili. Receptor cell or recipient cell or F minus cell or F minus strain not consider the sex pili. With the help of sex pili, these two bacteria are attached to one another. Conjugation tube is produced by sex pili. It is F plus and it is the F minus. Plasmid present in F plus strain replicates. It replicates. So it is not the semi-conservative method. It is a conservative method. Conservative method. Replicated plasmid or one copy that transfer to the F minus strain. F minus strain that receives that copy plasmid. So F minus strain, it becomes F plus strain. After conjugation, these two bacteria separate from one another. Both they consist the plasmid. Both they are F plus. It is also F plus. In case of the F plus strain, no loss, no gain of DNA or the plasmid. But in case of the F minus, it receives the plasmid from the F plus. So F minus strain becomes F plus strain. So genetic recombination occur. It receives the new genes. So genetic recombination occur by conjugation tube, and the two bacteria involved in this conjugation called conjugants. Conjugation tube produced by the conjugation tube. Sex pili. So genetic recombination or the transfer of genetic material occur in between two bacteria by direct contact in conjugation.